The first chapter in the unit reproduction deals with reproduction generally in organisms. The second chapter, reproduction in plants, and the third chapter, reproduction in animals. So, the outline of the events occurring in reproduction is in the first chapter. So, this is a brief outline of the events occurring in reproduction in general. What is given in yellow? The three events are pre-fertilization, post-fertilization and post-fertilization. So, the three events are pre-fertilization, second event is fertilization and the third event is post-fertilization. Now, pre-fertilization involves two processes. They are gametogenesis and gamete transfer. And we know what is gametogenesis. It is the formation of gametes from the gamete mother cell by meiosis. And the second event under pre-fertilization is gamete transfer. It is the transfer of gamete from the male sex organ to the female sex organ. Now, the second event is fertilization. As we all know, it's a universal rule. The male gamete fuses with the female gamete to form the zygote. And post-fertilization, the third event involves embryogenesis. That is, the zygote becomes the embryo. This is all in the first chapter, general outline. Now, second chapter deals with plants. We know pre-fertilization involves gametogenesis and gamete transfer. Here, the gametogenesis in plants are of two types because there are two types of gametes, the microspore and megaspore. So, accordingly, two gametogenesis occur in plants. They are microsporogenesis and megasporogenesis. So, two gametogenesis in plants. And the gamete transfer is called pollination. Then, that is in the first event, that is pre-fertilization. And pre-fertilization was already dealt in three slots. The first session I took was megasporogenesis. My second video was on microsporogenesis and the third was on pollination. So, today what I am going to take is the other two events, fertilization and post-fertilization. Fertilization. We know a brief recap of pollination. We know that pollen grains fall on the stigma. Once it falls on the stigma, a pollen tube germinates and this pollen tube penetrates to the stigma, style and ovary and enters the micropyle. And we know the pollen grain, a brief outline of the structure of the pollen grain, the outer exine, inner intine, and there are two cells, the vegetative cell and the generative cell. Nearly 60% of the angiosperm shed the pollen grains in the two-cell stage and remaining 40% of the angiosperms shed their pollen grains in the three-cell stage. So, which are the three cells? That is vegetative cell and two male gametes. As it falls on the stigma, the two male gametes, once the pollen tube is formed, it trickles down through the pollen tube and enters the ovule through the micropyle. Now, we are going to see what happens to these two male gametes. So, that is under fertilization. The first male gamete, I told you there are two male gametes. The first male gamete fuses with the egg to form the zygote. And this process is called syngamy. And the second male gamete fuses with the two polar nuclei to form the primary endosperm nucleus. This process is called triple fusion. This you all have learnt in the 10th standard. Because it involves two fertilizations, this process is called double fertilization. And double fertilization is only found in angiosperms, that is flower bearing plants. Which is the first fertilization? The male gamete fuses with the egg to form the zygote, which is called syngamy. And the second male gamete fuses with the two polar nuclei to form the primary endosperm nucleus. And this is known as triple fusion. These two fertilizations constitute double fertilization, which is unique to angiosperms. Next, so this is coming under fertilization. Next, we are going to see what is happening during post-fertilization, the third event, post-fertilization. So in post-fertilization, we are going to see the fate of the zygote and the primary endosperm nucleus. What are they destined to become? Now, what happens to the zygote is that it simply undergoes mitotic divisions to form various stages of the embryo. The first stage is the proembryo. The second stage, the globular embryo. Third stage, heart-shaped embryo. And the fourth stage is the mature embryo. So here you must remember the four stages the zygote will undergo. First is the proembryo stage, then it becomes a globular embryo, then it becomes the heart-shaped embryo, and finally it becomes the proper embryo, that is a mature embryo. These are the four stages the zygote undergoes purely by mitotic division. Meanwhile, what happens to the primary endosperm nucleus or PEN? I am representing it here in three structures. This is the primary endosperm nucleus, and from the diagram we can describe the structure. It has a single cell with a single nucleus. Now what happens? Look at the next structure. What has happened? What's the difference between the second structure from the first structure? Here you can see the same cell, but you can see a number of nuclei. So what must have happened is the nucleus alone underwent division, divided, redivided, and formed a multinucleated structure. This is not, the nuclear division usually is followed by cell wall formation, but here it is not followed by cell wall formation. So this structure is called free nuclear endosperm. So we have a multinucleated single cell. Next, you can see the third diagram. It is just the replica of first and second, but what's the difference is cell walls are laid. Look, the nuclei are there, multinucleated structure, and cell walls are laid finally. And this structure is called the cellular endosperm. So here the diagram is very important to remember the stages the primary endosperm nucleus or PEN is undergoing. So the first stage PN, it's a single cell with a single nucleus. Then what happens? The nucleus alone undergoes division, divides, redivides to form a multinucleated structure which is not followed by a cell wall. Such a structure is known as free nuclear endosperm. And finally the cell walls are laid around each nuclei to form the cellular endosperm. Now in the board questions they don't ask directly endosperm formation. Indirectly it is asked how is the free nuclear endosperm and the endosperm formed, final endosperm formed. And this can be seen in coconut. In coconut both the free nuclear endosperm and the endosperm are seen. The free nuclear endosperm is represented by the coconut water. When we open the coconut, there's a coconut water that is representing the free nuclear endosperm, and the coconut kernel, the white of the coconut, is the cellular endosperm. Coconut kernel that is used, you grate and use it in curries or we eat the edible part. That is the coconut kernel which represents the cellular endosperm. Now I have a question. Now I've drawn the two processes, that is zygote, what it is destined to become, and PEN, what it's destined to become. Two processes drawn simultaneously. Now the question I'm throwing across to you is whether these two processes are occurring simultaneously or whether the first process occurring first, that is embryogenesis occurring first, followed by endosperm formation, or the third option is endosperm formation is occurring first before embryogenesis. So once again, which process takes place first? First option is embryogenesis and endosperm development occurring simultaneously. That's the first option. Second option is whether embryogenesis is taking place first, followed by endosperm formation. And the third option is first endosperm formation followed by embryogenesis. Just think which should occur first. The event that is occurring first is endosperm formation. First, the endosperm, this occurs. Once this is completed, embryogenesis starts. This is very, very important because it's an adaptation to assure nutrition to the developing embryo. The embryo, as it grows, it requires nutrients. The nutrients are derived from the endosperm. So it cannot be simultaneous, or this cannot be after embryogenesis. Very, very important. What happens first is endosperm formation. So first the endosperm is formed, followed by embryogenesis. This is to assure nutrition to the embryo. Now moving on to the next topic. Now we see the embryo gets its nourishment from the endosperm. Here we have two categories of plants or two categories of embryos. Some embryos, mature embryos, utilize the entire endosperm. So they utilize the entire endosperm in such a way that the seed does not have any endosperm left. Such seeds are called as non-endospermous seeds. So what are non-endospermous seeds? The developing or maturing embryo, as they develop, they utilize the entire endosperm, leaving no residue of the endosperm in the seed. Such seeds are called non-endospermous seeds. Next, the second category of seeds. Some embryos utilize only a part of the endosperm, and the remaining bulk of the endosperm remains in the seeds. So such seeds are called endospermous seeds. So we have two types of seeds based upon this, whether the embryo is utilizing the endosperm completely or incompletely. If the embryo utilizes the endosperm completely, leaving no remnants of the endosperm in the seed, it is known as non-endospermous seeds. On the other hand, if the embryo utilizes only a part of the endosperm, leaving a bulk of the endosperm in the seed, then it is known as endospermous seeds. So I'm coming to the next topic, types of seeds, endospermous seeds, non-endospermous seeds and endospermous seeds. As I told you, non-endospermous seeds, the entire endosperm is utilized by the developing embryo, and endospermous seeds, only a part of the endosperm was utilized. The remaining endosperm remains in the seed. See what I have highlighted in yellow is the endosperm. And we have learnt in 11th standard which seed is this. This is a dicot seed and this is a monocot seed. So remember, non-endospermous seeds example are the dicot seeds, and the endospermous seeds example are the monocot seeds. Now, a brief look into the structure of an embryo. An embryo, whether it is a dicot or monocot, has an embryonal axis. The upper part of the embryonal axis is called epicotyle, and the lower part is called hypocotyle. The epicotyle gives rise to the plumule, which gives rise to the shoot system, and the hypocotyle gives rise to the radicle, which finally gives rise to the root system. So an embryonal axis has the upper portion called the epicotyle and the lower portion called the hypocotyle. It's common to both the seeds. Now, in dicot seed, there is no endosperm. Instead, for germination of the seed, the nourishment is taken from the cotyledon. So they have two fleshy cotyledons from which they take the nourishment for development. On the other hand, look at the endospermous seeds. In the endospermous seeds, the cotyledon has no role. It's a single cotyledon. It's flat like a shield, pushed towards the corner. So this is the cotyledon. It's flat shield-like, and this is called scutellum. We have learned in 11th standard. No role. All the nourishment is again derived from the endosperm. This is the endosperm. Now I'm placing the embryonal axis in black color. This is the embryonal axis. The embryo located on the lower side of the scutellum. It has a typical hypocortile and epicortile. And remember, here there are additional structures. The hypocortile is protected by a sheet called chorioryza, and the epicortile is protected by a sheet called choriotile. All these terms we have learned in the 11th standard. Anyway, I'm going to write it. And this is the choriotile. So that's the structure of the endospermous seed. We are going to look into the advantages of seeds to angiosperms. The first advantage of seeds to the angiosperms is that it is, we saw what and all events were involved to form the seed. We saw pollination, we saw fertilization, all were independent of water. So thus seed formation is a dependable process. It does not require water. Any events preceding the final seed does not require water. Events like pollination, fertilization does not require water. So seed formation is a very dependable process. Second advantage of seed is they have adaptive strategies for seed dispersal. Right from lower classes, you'll have learned how seeds are dispersed. They have wings or tuft of hair or hooks, etc. That's how they're carried from the parent plant to distant places. So because of this adaptive strategy, they are widely distributed. The third advantage is all seeds are protected by a seed coat. So this protects the young embryo. The young embryo is not going to die easily. It is protected by a hard and tough seed coat. And lastly, and very importantly, seeds are the product of sexual reproduction. And we know if it is a product of sexual reproduction, there is combination of characteristics of two parents, which means it results in variation. And we know the advantage of variation. Survival of species. So that's the fourth advantage. So a quick recap of the advantages of seeds to angiosperms. Seeds is a reliable process because pollination or fertilization is independent of water. Second, seeds have adaptive strategies so that it can be dispersed to distant places. Third, seeds are protected by seed coat, and therefore the young embryo is not dehydrated. It is well protected. And lastly, it is a product of sexual recombination or sexual reproduction. Therefore, there's recombination of characters. Therefore, there's variability and helps in the survival of the plant. Now we are going to see the next topic is viability of seeds. Viability of the seeds means how alive the seeds are. Once they are dispersed, will they germinate? So that is known as viability, the life of a seed. Not all seeds are viable. It varies. The duration varies from species to species. Some species, the viability lasts for some months, for some for minutes, and some it should be, once dispersed, it should be immediately sown. So we're going to see the viability of two rare seeds where the viability existed for long. The two cases is, the first case is Lupinus articus. The viability lasted for 10,000 years. This was found by when the ice caps in the tundras were excavated. They found this particular seed. They sowed the seed, it germinated and flowered after 10,000 years. See the length of the viability, how long it was viable. The second example is Phoenix dactylifera. It's a date palm seed which was found by the archaeological department when they excavated the palace of King Herod near the Dead Sea. We have heard about King Herod. It has reference to the Bible. And in his palace, this date palm seed was excavated and it was germinated. It was sown and germinated and it flowered. And its viability lasted for 2,000 years. So we have two cases of long-lasting viability. One is Lupinus arcticus from the, as the name suggests, from the Arctic tundras, and the other one is Phoenix dactylifera, which was excavated from the palace of King Herod. This one's viability lasted for 2000 years, and here this one very long lasting viability, the duration it took place 10,000 years.